chapter in our biochemistry lecture series in our Medicos app. Here we'll be learning about plasma proteins. Plasma proteins are nothing but proteins present in the plasma and they have their own varied functions. Plasma proteins. First we need to study about various plasma proteins and their functions various plasma proteins and their functions. Now, most important plasma protein that is there is our transport proteins. Transport proteins that are required for transport, like here albumin transports almost everything. Then your transferrin, which transports iron, ceruloplasmin. Then we have transcobalamin, cortisol binding protein, sex hormone binding protein, retinol binding protein, prealbumin or transthyretin. So some of the various transport proteins. Then we have acute phase proteins, positive and negative. Acute phase proteins. Most important of it is C-reactive protein. C-reactive protein, which is elevated in almost all infections. Then alpha-1 antitrypsin, alpha-1 acid glycoprotein, also called as orosomucoid, their ceruloplasmin, etc. So, acute phase proteins are those proteins which get elevated during acute inflammations. Alright. Then there are various classifications of functional and non-functional plasma proteins. Functional and non-functional. Functional and non-functional. Functional proteins are those which are having a proper function inside the blood. Like say clotting factors. They have work inside the blood. They do the clotting. Fibrinogen, the protein which is having a function inside the blood. So these are functional proteins. Non-functional protein like your lipase. Lipase has no work in the blood, but it is there inside the blood. Amylase, SGOT, SGPT, alkaline phosphatase, all these have absolutely no work in the blood, but they are still present inside the blood. So these are non-functional. They are just hanging out, no work. Who is exactly doing the work? These clotting factors, fibrinogen, etc. These are the ones that are doing the work. Alright. Apart from this, plasma proteins, most important part that you need to study is the electrophoretic pattern. Electrophoretic pattern. Now this electrophoretic pattern in different diseases, that you'll come across in your spotters, your objectively structured practical exam, you will be facing an electrophoretogram. This electrophoretogram will be mostly of your plasma proteins and they will be checking your knowledge whether you know all of them or not. So electrophoretic pattern, basic pattern on an electrophoretogram is a big gamma band followed by beta band followed by alpha band and followed by albumin. Okay. So this band is albumin, alpha band, beta band and a gamma band. Of course, there are alpha 1, alpha 2, beta 1 and beta 2 bands also. So albumin will be something like this. or. Okay, so that will be your albumin. Now, in specified cases like multiple myeloma, monoclonal antibodies will be increased and you'll get a monoclonal antibody band here in gamma region. In nephrotic syndrome, liver disorders, various other conditions, chronic inflammation, these electrophoretic patterns change based on which protein is elevated. Okay, based on which protein is elevated. So this electrophoretic pattern is a must know, very, very important for you to remember. Which band, gamma band is having which proteins, beta band is having which proteins, alpha band is having which proteins. Your alpha 1 acid glycoprotein, alpha 1 antitrypsin, alpha 1 microglobulin, beta macroglobulin, ceruloplasmin, transferrin, all of LDL, all this will occur here. Albumin and pre-albumin band, your fibrinogen, albumin and pre-albumin or transthyretin will occur in this band. Alright, so you need to make a mnemonic. We have discussed in the class what each band contains and what are the different types of electrophoretic patterns that are going to come in your examination. Alright. So that is about plasma proteins. You need to remember what are acute phase proteins, what are transport proteins, and also a hell lot about albumin. Read everything about albumin. We have discussed in great detail about albumin. It is 65,000 kilodaltons. What does it carry? How does it measure? How does it maintain the oncotic pressure inside the blood? How deficiency of albumin will lead to edema? All this has been discussed.